All right, let's talk about chemistry in biology, which would fall under the categories of organic chemistry and biochemistry. Organic chemistry means something different than you might think just from the word organic. So normally we would think organic would mean something that's natural or something that is found in living things. But in chemistry, organic just means it has carbon in it. So everything that has carbon would be covered under organic chemistry, even things that are completely unnatural like polyester or acrylics or plastic. Those would be considered organic chemicals because they contain carbon. There's a reason why we are a carbon-based life form. Carbon is the only element on the periodic table that can form very long, stable chains at the temperature at which water is a liquid. So carbon is the basis for all of our biologically important molecules, proteins, which we'll talk about, all of these lipids, carbohydrates, and nucleic acids, which nucleic acids would be DNA and RNA. So carbon is the basis of the structure of each of these. The only important molecule that's not based on carbon is water. And because water doesn't have carbon, it is not considered an organic molecule, even though it makes up 70% of our bodies. So here's our four most common elements in biological molecules again. Hydrogen, which forms one bond. Oxygen, which forms two bonds. Nitrogen forms three bonds, and carbon forms four bonds. And nitrogen and oxygen, remember, are very electronegative. So whenever you see oxygen and nitrogen bound to hydrogen especially, but also to carbon, they're going to have a partial negative charge, and that's going to help whatever molecule they're a part of be hydrophilic. Biological molecules have a lot of the same patterns that we see over and over and over again, and what makes them different is what we call functional groups. So the functional groups are molecules that could increase whether a molecule is hydrophilic or hydrophobic, or affect what receptors in the body these molecules can bind to. And just as an example here, so the yellow structure here, sorry about that. So, oh, I need my pointer back. So these molecules here are based on cholesterol. The yellow structure is a cholesterol molecule. Cholesterol is hydrophobic, and if you forgot about what these line drawings mean, so because they're in organic chemistry, there are so many carbons and hydrogens, we can reduce the carbons to mere points. So every point, every corner on this ring structure is a carbon. So carbon, 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 carbon. And we just assume all the hydrogens that are needed to make carbon, all the carbons have four bonds. So there's some double bonds here. That's what the double lines are. So this carbon right here at this corner, it has three bonds already, and we just need one more bond to make four bonds. So we're going to assume that there's a hydrogen on here, just to keep everything from being completely cluttered with hydrogens, which it would be if we drew everything out. So these two molecules, these are both steroid hormones. Steroid hormones are all manufactured from cholesterol in your body. Cholesterol is such an important molecule that you can make all the cholesterol that you need. Estrogen, estradiol is one form of estrogen. There are several slightly different forms, and testosterone. And you can see there's very little difference between these molecules. We have one extra hydrogen over here, and we have a methyl group here. And that makes the difference as to which receptors these molecules can bind to. So there's estrogen receptors and there's testosterone receptors, and they don't bind to each other's receptors. So functional groups are very important for the shape of the molecule and also for the properties of the molecule, like does it have a charge, can it dissolve in water, etc. So here's some of those functional groups. These are small groups of atoms that are attached to these larger carbon-containing groups. That's a functional group. A functional group by itself is not a molecule. A functional group is part of a larger molecule. So the first one here, a hydroxyl group, this one is just an oxygen and a hydrogen that's bonded to a larger group. And we see these a lot in carbohydrates. For instance, glucose molecule has one of these on every carbon in the glucose molecule. It's also present in alcohols like ethanol. So this is ethanol. It has 2 carbons and our 1 hydroxyl group. So, and because of the oxygen being electronegative, a hydroxyl group is going to help increase the likelihood that a particular molecule is going to be hydrophilic. So alcohol is hydrophilic because of this oxygen here. It also has hydrophobic properties as well, which, you know, because nothing is simple in biology. There are molecules that can both dissolve in fat and dissolve in water, and ethanol is one of those. The carboxylic acid group is a weak acid. It's not as strong as, say, hydrochloric acid, which would be a strong acid. And a carboxyl group is a carbon bonded to 2 oxygens with only a single hydrogen. And the way we draw it here with 1 double bonded oxygen and 1 single bonded oxygen, actually the carbons share this hydrogen, and each oxygen has a partial negative charge as well. And because it's an acid, remember that acids have an excess of hydrogen ions, so it tends to lose that hydrogen. So that's what makes it an acid. And because it doesn't completely dissociate, that's what makes it a weak acid. So vinegar, which is a very common organic acid, it is formed by bacteria from ethanol. It has 2 carbons, and one of those carbons is a carboxylate ion. So we sometimes just call this an acid, an organic acid, or if you want to be more correct, carboxylic acid. Amino groups also very important, and again, like a carboxylic acid group or a hydroxyl group, amino groups are going to increase the molecule's ability to dissolve in water. Because this nitrogen is electronegative, it's going to have a partial negative charge. And here we have one of the types of molecules where we find amino groups, and that's amino acids. So amino acids are called that because they have an amino group and they have an acid group. So here's our simplest amino acid, glycine. It has an amino group and it has a carboxylic acid group. And you'll notice that it doesn't matter which direction we draw these oxygens. That doesn't matter because this molecule can flip around in any orientation and it's still an amino acid. So, and it's basic. Amino groups are basic because they tend to suck up a hydrogen from the watery media around them and become a positive ion, and so that's why they are base. So amino acids don't affect the pH because they have both a basic end and a acidic end, unless they have some other group on there that affects it. So amino acids all have these 2 groups, an amino group and a carboxylic acid group. Phosphate is a really important functional group. Phosphates are part of DNA and RNA. And phosphate is weird. So a phosphate is a phosphorus with oxygens around it. And phosphorus, if you look at the periodic table, phosphorus is right below nitrogen. So we might expect it to have similar properties to nitrogen. We would expect it to make 3 bonds like nitrogen does, but in fact it doesn't. Phosphorus behaves weirdly and can form 3, 4, 5 bonds. But that helps with the other properties that it's important for. It's also part of the structure of ATP, which is an important energy molecule, which is a nucleotide. We will talk about those after we talk about proteins. But it's highly charged. As you can see, phosphate groups are really going to help a molecule dissolve in water because they're highly hydrophilic because of all these oxygens and their negative charges. A methyl group is just a methane that's missing that last hydrogen, and instead it's stuck onto a larger group. So here we have cytosine, and methyl group is stuck on here. And you'll notice that even though this has 3 hydrogens, we don't draw out all the bonds. We just write it as CH3, and that's another shorthand way that we represent things in organic chemistry. Here's another one here. Nitrogen is bonded to 2 hydrogens, and instead of drawing them out with their little bonds and 2 H's up here, we just draw the amino group as NH2, and that's something that you'll commonly see. And often these hydrogens down here will just be left off because we get tired of drawing all the hydrogens. So a methyl group, because it doesn't have any oxygens, nitrogens, or sulfurs, is also going to be hydrophobic. And if a molecule has a lot of these, that's going to help at least that part of the molecule to be hydrophobic. So for organic chemistry then, we're a carbon-based life form. All of our important biological molecules, with the exception of water, are based on carbon. And it's because carbon is such a versatile molecule. It's the only element in the periodic table that can form huge, long, stable chains, millions of carbons long, at temperatures where water is a liquid. And so biological molecules are all based on these carbon chains. Proteins, carbohydrates, fats are all, and nucleic acids like DNA are all based on carbon. And we'll talk about each of those 4 major groups. And then the functional side groups like amino groups and carboxylic acid groups and hydroxyl groups, those change the properties of whatever that carbon chain is going to do. So in biology, we have what we call macromolecules. So macro means big, so big molecules. The biggest molecules in chemistry are in biology and biochemistry. And 3 of the 4 categories that we're going to talk about are what we call polymers. So a polymer is made up of long chains of repeating subunits called monomers. Mono means one, poly means many. So a polymer is a long chain of repeating units. Those repeating units are monomers. So for each of the categories of molecules that we're going to talk about, think about what we call the polymers, because we have different names for them, and what the monomers are and what the functional groups are in each of those monomers. The only group that doesn't have polymers and monomers are the lipids. All the rest, the proteins, carbohydrates, and nucleic acids all have monomers and polymers. Proteins are made of amino acids, which you might already know. You've probably heard of the 10 essential amino acids. Those are the amino acids that we cannot make, that we have to eat in our diet. And the reason we need those amino acids is to build proteins. There are 20 amino acids total that we need to build our proteins, and 10 of those are considered essential amino acids because we cannot manufacture them. Most of the other amino acids that we can make, we make from other amino acids, and a few of them we can build from scratch. So here's a representation here of links of amino acids. So you'll notice each amino acid has a symbol, and we have 3-letter abbreviations for all the amino acids, and we also have 1-letter abbreviations. We're not going to use the 1-letter abbreviations, but you don't have to memorize them either, but you should recognize them. If I say glycine, isoleucine, valine, cysteine, glutamine, you should think, oh yeah, those are amino acids. If it's a short chain, we usually don't call it a protein, we usually call it a peptide. So if it's less than 100 amino acids long, we usually just call it a peptide. And there are many functions in our cells. Your cells make over 20,000 different proteins. And some of them are structural, like collagen. Some of them are enzymatic, meaning they make particular reactions go faster. Some of them regulate your genes and determine which genes get turned on and off. Some are part of the immune system. Some are signaling molecules between cells. We've heard about some of these with COVID, like the cytokine storm. If you've heard of that, that involves proteins that are made by immune cells. And the amino acid sequence is going to determine what the function is. So we can look at the amino acid sequence of a particular protein and sometimes figure out quite a bit about what that protein might do. So amino acids all share a basic structure. So that there are millions of possible amino acids. If the definition of an amino acid is just a molecule that has an amino group and a molecule, and it also has a carboxyl group. If that's the definition of an amino acid, then we can think of a million possible amino acids. But in reality, there are only 20 that are used by animals, and then there's a couple of extra weird ones that are used by bacteria that we don't use. But they all have basically the same structure, the 20 that we use. And they have an amino group, a central carbon, and a carboxyl group. And that central carbon that's where the variation is in the 20 amino acids. What's in this side chain? The simplest one just has a hydrogen, that's glycine. Alanine just has a methyl group. Tryptophan is a complex one that has a ring structure here with a nitrogen in it. So some of them are quite complex and some of them are very simple, but that's the part of the molecule that varies, is this side chain. But because of this amino group and this carboxyl group, all the amino acids by themselves are hydrophilic. It's only when we link them together into proteins that we can create regions that are hydrophobic or hydrophilic. So these are some of the amino acids that have hydrophobic side chains. Now that doesn't mean that the amino acid by itself is going to be hydrophobic. All amino acids are hydrophilic alone, and they will dissolve in water, which is fortunate because all of the protein synthesis that happens in our cells happens dissolved in water. It would be quite difficult if any of the amino acids were not hydrophilic by themselves. But the side chains here, which is the yellow, the purple is what's the same in every amino acid. The yellow side chain is different. So you see several of them just have carbons and hydrogens, and that tells us that that side chain is going to be hydrophobic. And what that means is if we link together a bunch of hydrophobic amino acids into a peptide or a protein, this part of the molecule where we find these hydrophobic side chains is going to repel that watery environment inside the cell. And often that means that's going to fold to the inside of the protein, and it's going to help the protein to fold up correctly. So here are a few other ones. You see one here has sulfur. This isn't enough, however, to make it hydrophilic. The same with tryptophan. This one little nitrogen here is not enough to make this side chain hydrophilic. Some side chains are polar, like serine, threonine, cysteine, etc. And that's because you'll see oxygens, sulfur bonded to a hydrogen, amino groups. Those help these to dissolve in water. Some of the side groups on amino acids are actually acids or bases. And these are also going to help proteins to fold up correctly because the negatively charged acids are going to be attracted to the positively charged amino groups, and that's going to cause these to stick together during protein folding because all of our proteins, they start out as a long linear chain of amino acids, but they all fold up into some three-dimensional shape, and that's what determines their final function. The umbrella term for all of these molecules that are made of amino acids is polypeptide. And the peptide does not refer to the amino acid. It refers to the bond that forms between amino acids. So when the amino group on one amino acid bonds to the carboxylic acid group on another amino acid, that's a peptide bond. And if we were naming them today, perhaps we'd name them polyamino acids. But the biochemist who named them in the 1800s, named them polypeptides, and so we're stuck with that. So polypeptides can be quite short. They can be only a dozen or so amino acids, like peptide hormones, like insulin is a peptide hormone, and it's quite short, to thousands of amino acids, like some larger proteins. And here's another one of those. Proteins that's represented by each amino acid is a little bead in this case. That's one of the ways that we draw proteins. So a peptide bond forms when an amino group and a carboxylic acid group form a bond. And this is called dehydration synthesis because a water molecule is lost during the process. But what I want you to notice is that this makes the backbone of our protein nitrogen, carbon, carbon, nitrogen, carbon, carbon, nitrogen, carbon, carbon. And we are going to do one of our lab experiments for the detection of the presence of protein is going to be based on this nitrogen. We have a test called the Biuret reaction that binds a copper ion to this nitrogen, and so it can detect the presence of protein because of the presence of this nitrogen in roughly the same amount relatively in every protein. It's going to have about the same proportion of nitrogens. The amino acid sequence and whether there are hydrophilic or hydrophobic side chains could tell us something about the function. So, for example, if we discover a new protein, we have no idea what it does. So here's this brown snaky thing here. That's one long polypeptide here. And if we notice that there are long regions that are all hydrophobic side chains, that's a good indication that perhaps this protein sticks in the membrane. So that's one example of how we can use the amino acid sequence to help us predict the function. Long hydrophobic regions often indicate a protein that ultimately is going to stick in the membrane because the inside of the membrane, as we'll see, is also hydrophobic. Unlike the rest of the watery outside the cell, watery inside the cell, the fatty part of the cell, the hydrophobic part of the cell is inside of the membranes. Proteins can also be enzymes, and this is another way that we represent proteins by using this ribbon structure. And in this case, we can't see the individual amino acids at all. All we can see is the overall shape. And so this is an enzyme here that's made up of multiple polypeptide chains, each color, like the green, the purple, pink, yellow. Each color is one polypeptide, and it requires several of these polypeptides together to make the functional enzyme. And an enzyme can either break down molecules into smaller pieces, like this one, proline degrading enzyme, or it can assemble things together, like DNA polymerase is another enzyme that's made out of protein. Enzymes can catalyze all sorts of reactions in the cell. In fact, pretty much every reaction that occurs inside of your cells is catalyzed by some kind of an enzyme, a protein enzyme. And this is an example of an enzyme that one of our labs uses. This is the enzyme catalase. It's found in many of our mitochondria-containing cells because hydrogen peroxide is a byproduct of oxygen metabolism, and it's not good to have around in your cells. So we need an enzyme that will break it down almost as soon as it forms, and that's what catalase does. It catalyzes reaction. So to catalyze a reaction means to make a reaction go faster. Proteins can serve as transport molecules. So this is a cell membrane here with the gray in the center here being the hydrophobic interior of the membrane. And anything that's water soluble, including water, cannot cross that fatty membrane. So we have to have transport molecules to get things across the membrane. And we'll talk more about those when we get to the membrane section. This is one example of such a molecule, and pretty much all cells on Earth have some version of this protein called aquaporin, which aqua means water in Latin, por is a hole in Latin, so aquaporin literally means a hole for water in the cell membrane. And it allows our charged water molecules to go back and forth because they could not cross this hydrophobic membrane without that kind of help. We also use protein to transport things around in our blood. Ferritin is one example of that. Ferritin has 8 polypeptide chains and of 4 different types. So the blue ribbons here are one type of polypeptide chain. The purple ribbons are another type of polypeptide chain. And ferritin, as you might be able to tell from the root here, ferrous, which means iron, that it transports iron around. Iron is very reactive and would be catastrophic to have loose iron floating around in your bloodstream. That would be really bad. So the way iron gets from your intestine to your liver and then to other cells that need it is by being carried on a protein. And so a ferritin protein molecule can carry iron around in your body safely without it wreaking havoc on the insides of your blood vessels as it goes. Proteins can also be structural. One of the most important structural proteins in our bodies is collagen. This little slice here, this is an electron microscope image from a bone. And all the cells have been digested away. Your bones are living, they have living cells in them. You can kind of see the holes where the cells were here. And what's left is the collagen. And it forms very strong, ropey fibers, but yet it's just made out of amino acids. It's just a protein. And the different colors here are different polypeptide chains. They're all the same. They're just colored so you can see the kind of braided structure that collagen has. Collagen is actually what holds our bones together. We like to think of our bones as being made of calcium, but calcium is just a hardening agent. Most of the structure of our bones is made of collagen, and that's why people who have protein deficiency, osteoporosis is often one of the first effects of protein deficiency. It's because protein is collagen, and if you can't make enough protein because you're not eating enough protein, you're going to have problems making bones, and you can end up with weak bones because of that. So your bones are mostly made out of protein. Protein is also the skeletal elements inside the cells, which we call cytoskeleton. Cyto means cell. So the cellular skeleton, here are two cells here. And what we can see here is two of the cytoskeletal protein elements that are lit up with different colors. So microtubules and actin filaments, which we'll talk more about when we get to the cell chapter. The microtubules are the yellow fibers here, and the actin filaments are the purple, which are around the edge of the cell. These are both made out of protein. They're different proteins, but they're very important for the cell structure and shape. We also use proteins to defend ourselves. We've been hearing a lot with the coronavirus about neutralizing antibodies, and we want to make sure that the vaccine is generating the production of antibodies in our bodies. Well, the antibodies are proteins. Antibodies are proteins that are made by specific immune cells that match the shape of some foreign invader that it's attacking. And it could be the surface of a bacterial cell. It could be the surface of a virus. And that's what antibodies do. And so when you have the ability to make an antibody that exactly matches some invader, your body will make it in very large quantities. And that's what vaccines trigger, is they trigger all of the matching antibodies to whatever the vaccine has in it, whether it's a flu virus, whether it's a tetanus bacteria, whatever the antigen is in the vaccine, it's going to trigger the production of antibodies that can bind to that and hopefully disable it, which is what a neutralizing antibody is, because you can make antibodies that stick to something that don't neutralize it. So we want neutralizing antibodies. So that's just another type of protein that our bodies make. Proteins are important in muscle contraction, and I'm just showing you this as an example. I don't want you to memorize the details of this. If you're going to go on and take physiology, you can learn a lot more about muscle contraction there. Muscle contraction involves two important proteins, actin and myosin. Actin is that same one that I just showed you that also is part of the cytoskeleton inside cells. It's also part of muscle contraction. And these little purple beads here, those are actin filaments. And myosin is a different protein that walks on the actin. So when you contract your muscles, the myosin protein walks along the actin and actually makes the muscle cells shorter, and that's a muscle contraction. Gene regulation as well is also all about proteins. We have many different types of proteins that bind directly to genes and turn them on or turn them off, and they are mostly different kinds of proteins. This is one example. The little brown thing here is DNA. You can kind of see the little double helix there. And the green is a protein in the ribbon structure binding to that DNA. Our DNA is also organized around protein. These little colored balls here are histone proteins. And our DNA is so long that it needs to be organized by winding it around these types of proteins. And so this is one level of DNA structure, is proteins. So the first level of structure of a protein is just what amino acids are there. And we call that the primary structure of a protein. The primary structure is just the sequence of amino acids. This is not affected by cooking, by pH. These are all covalently bonded together with peptide bonds. It's very stable. You need an enzyme to break those bonds. The next level of protein structure involves folding. And there is hydrogen bonding even within the amino acids in a single polypeptide chain. And these amino acids will hydrogen bond together and form two basic kinds of structures. One is a beta pleated sheet and the other is an alpha helix. And you see here the Greek letter, this is lowercase beta and a lowercase alpha, which kind of looks like a sideways breast cancer ribbon. You could write out the word beta, B-E-T-A, or alpha, A-L-P-H-A, if you don't feel comfortable writing the Greek letter. And we call that the secondary structure. Not all proteins are going to have secondary structure. If it's a short peptide, it might not have any secondary structure. But most proteins, if they're long enough, will have some helical structures to them. That will be alpha helix. And many also have beta pleated sheets. So we call that, if it's present, secondary structure. And finally, the tertiary structure is how one polypeptide chain folds up. All proteins have some tertiary structure, some three-dimensional structure that's important for their final functioning. And the way that they fold up into the tertiary structure involves ionic bonding, so ionic bonding between those acid and basic side chains. And also, here's another even weaker kind of bond, hydrophobic interaction. So we had ionic bonding, covalent bonding, which was shared electrons, and then hydrogen bonding, which is even weaker. So hydrogen bonding is weaker than covalent bonds or ionic bonds. And then even weaker than that is hydrophobic interactions. So what hydrophobic interactions are, it's like if you've ever had a bottle of salad dressing, Italian salad dressing or Greek salad dressing with the oil and the water layers. You shake it up and the oil breaks up into tiny, tiny little droplets and is dispersed throughout the water. But as you let it sit, all those oil droplets touch each other and fuse. That's because of hydrophobic interaction. So in a watery environment, which everything in life is a watery environment, inside our cells, outside our cells, it's all a watery environment. And anything that's not hydrophilic, all the hydrophobic things are going to want to stick together because they repel the water. And that's what happens inside of these three-dimensional structures. So if we have any amino acids with hydrophobic side chains, those hydrophobic side chains are going to be repelled by the water and they're going to end up in the inside and they're all going to glob together like one big oil droplet. And then finally, if the protein has more than one peptide chain, which many of them do, like a couple of the examples that I showed you, then it has quaternary structure or fourth level structure. So not every protein has this. Some proteins, like catalase enzyme that we're going to use, is only a single polypeptide chain, so it does not have quaternary structure. But if it's made up of more than one polypeptide chain, then it has quaternary structure. Hemoglobin is an important one. We're going to use hemoglobin as an example in a few different chapters here. Hemoglobin is found in your red blood cells. It helps carry oxygen around in your blood. And it's made up of 4 different polypeptide chains, 2 alpha chains and 2 beta chains. And it also has an extra little molecule here called a heme ring or a porphyrin ring. And the heme ring doesn't fit It's a carbon-based molecule, but it doesn't fit into one of our 4 categories. It's not a protein. It's not made of amino acids. It's not a carbohydrate. It's not a lipid. And it's not a nucleic acid. And this is an example of another type of molecule that's used in smaller amounts, doesn't fit into one of those big categories. So there are a bunch of smaller types of molecules that are enzyme cofactors and other things that, like heme, that are added to proteins or added to other molecules or added to fatty acids that don't strictly fit into one of those 4 categories. So heme is what the heme group, the heme ring is what holds on to the iron atom, and that's what allows it to carry oxygen. Now, if one of these amino acids is not correct, this protein isn't going to fold up correctly. And that's going to be a problem for whoever has that particular molecule. So when proteins become misfolded, we call that process denaturing. So when you denature a protein, that means you put it in an environment where it doesn't stay in its tertiary and quaternary structure. It unfolds. Now the primary structure is unaffected. The primary structure is just the sequence of amino acids, and you have to have an enzyme to take that apart. Denaturing is, you could do it with high temperature, with cooking. You can do it with high salt. You can do it with acid pH or very alkaline pH. And that's in fact why the texture of food changes when you cook it, when you pickle it, which involves high salt, or if you put an acid on it like lemon juice, or if you put a base on it like lye, like in some fish preservation methods. So generally, when you denature a protein and you destroy its tertiary structure, it isn't going to go back. So when you cook food, when you cook meat, let's say, you denature all of the muscle proteins that are in there. When you cool the meat back down, it doesn't go back. Everything stays denatured. When you cook beans, for example, you denature all the proteins in there. That's why they change texture. When you cool them off, it doesn't go back to the texture that it had before. So denaturing is usually irreversible. The thing that does not change is the primary structure, the amino acid sequences. The sequence of amino acids is still there. It's just the folding has been destroyed. That's what denaturing is. And it generally destroys the function of the protein. So if I have an enzyme like catalase and I boil it, it's not going to work anymore. It's because its function depends on that 3-dimensional shape. There are also diseases associated with misfolded proteins, Alzheimer's disease and mad cow disease, but there's new ones being added to the list all the time. Parkinson's has been added to this list as well. We now know that in Alzheimer's disease, there are 2 types of proteins that we find in the brain that are misfolded. One is outside the brain cells, and that's amyloid B protein, which you might have heard of. And the other one is inside the brain cells, another protein called tau protein. We find it in the brains of Alzheimer's patients, misfolded and glommed together in long strands. And at this time, though, this observation has not led to any effective treatments for Alzheimer's. Mad cow disease, similarly, and humans can get mad cow disease as well. Mad cow disease and Creutzfeldt-Jakob disease is what it's called in humans, are caused by prions. Prions are misfolded proteins, and normal prion protein is present in all mammal brains, including human brains, mouse brains, dog brains. We don't know what the normal protein does. It's there in all of the mammal brains, but we do know if you misfold the protein, so you didn't change the amino acid sequence, you just fold it wrong, that misfolding becomes contagious and it will misfold its neighbors. So here on the left is a prion protein with its normal folding, and you can see the nice alpha helices here. If it's misfolded, and this is called the rogue conformation, it's the same amino acid sequence, but now it has more beta sheets and less alpha helix. This misfolded prion can touch other prion proteins and misfold them too. And that's what happens in the brain, is that this protein gets into the brain, usually through the diet, and gets into the brain and starts slowly causing a chain reaction of protein misfolding in the brain that eventually results in brain disease, and that can take months or years to display itself. There are many, many, many genetic disorders that are the result of a protein that is misfolded because the amino acid sequence is altered. And one of those is sickle cell disease. And we know the most about genetic diseases that happen to be common in certain populations. And sickle cell disease is one of those. We're also going to talk about cystic fibrosis and a few others that are also common in particular populations, because that makes them easier to study if you have lots of individuals to study. Very rare diseases are more difficult to study. So in sickle cell disease is a recessive genetic disorder, so that means you have to have 2 copies, one from each parent, of the right allele in order to have the disease. And it results from a single change of one amino acid in one of the hemoglobin genes. So hemoglobin has 2 different peptides, or polypeptides, alpha chains and beta chains. And in individuals who have sickle cell anemia, they have a single mutation in only the beta globin gene. So normally hemoglobin here with its 4 little subunits forms this nice little tertiary structure, fills up the red blood cells. In individuals who have, well, if you're a carrier, you have 1 gene for this, but you won't have the disease. When the beta globin forms, it has a valine here instead of a glutamine, and this one is hydrophilic, valine is hydrophobic. So what it does is, when this beta subunit here folds up, we end up with a small hydrophobic region that is repelling the watery environment around it. And if you have enough of them near each other, these hydrophobic beta globins are going to be attracted to each other by hydrophobic interactions, and they're going to stick together. And they stick together and form these long sheets, and that deforms the red blood cells and causes them to break. And it's the breaking of these red blood cells that causes the symptoms that we associate with sickle cell disease, which even with excellent medical care, it is still a terrible disease to have. It requires regular hospitalizations. It's painful, blood transfusions. We don't have good medical treatment for it. It looks like gene therapy is going to be the best treatment for sickle cell disease. And hopefully that is coming soon. So that's the end of the protein section. And in the next video, we'll talk about nucleic acids like DNA, carbohydrates, and lipids.